All right. It's 5:15, so in interest of time, we'll go ahead and get started. Good evening, everyone. I'm Manmeet Alavali. I'm a medical oncologist, and I serve as the chair of the CNS Working Group. We have a fairly intimate group here. I'll probably start with Dr. Chad Rustovan, who is going to talk to us about some of the trials that he's been championing for SWOG and presented to the group. Chad.
Thank you, Manmeet. It's a pleasure to get to present to the group. So I have a couple of trials. The first one that I'll talk about is a trial that was actually developed through SWOG via the lung group. And then I'll talk about some other trials that are endorsed by the SWOG CNS Working Group. So this first trial is the SWOG S1827 trial, or so-called MAVERICK trial. This is MRI brain surveillance alone versus MRI surveillance and prophylactic cranial radiation, a randomized phase 3 trial for small cell lung cancer. And there's our study team. And so for just brief background on this question, prophylactic cranial irradiation, or PCI, in small cell lung cancer has historically been associated with a decrease in brain metastases, but an increase in neurological toxicity. And there was a lot of debate about its use in the 70s and 80s and then early 90s, and it became the standard of care following two landmark studies that were both published in the New England Journal of Medicine, showing an improvement in overall survival. So the first one was a meta-analysis of trials in limited stage, published in 1999, and the second was a dedicated randomized trial in extensive stage, published in 2007. And though they both showed a benefit in terms of overall survival and made PCI the standard of care, the studies were noted to have some limitations, including heterogeneous or absent brain staging and surveillance imaging. And so the question was, are we missing some patients that had brain metastases up front? And are we intervening too late because we're not doing rigorous surveillance? Subsequently, there's a Japanese randomized trial in extensive stage published in 2017 that specifically looked at this type of a question, where all patients had staging and rigorous brain MRI surveillance, and then they were randomized plus or minus PCI. And in that randomization, there was no differences in progression-free or overall survival. And so the implications of this were that MRI surveillance, perhaps allowing early salvage therapy for patients who develop brain metastases, may allow for the avoidance of PCI and its toxicities. And the NCCN, in response to that data, categorized PCI as optional for patients with extensive stage and began to recommend MRI surveillance for all small cell lung cancer patients. And so that leads us to today, where we have this question where we don't know what the role of PCI is in the modern era of brain MRI surveillance, particularly in patients with limited stage small cell lung cancer. But also we now have conflicting data in extensive stage small cell lung cancer, one trial showing an overall survival benefit and one trial showing no benefit. And so we designed this trial, the MAVERICK trial, to address those two questions, to be the first trial of MRI surveillance plus or minus PCI in limited stage, and to be a tiebreaker trial in extensive stage. And so it's a straightforward trial, one-to-one randomization for patients with small cell lung cancer to PCI versus no PCI. All patients get rigorous MRI surveillance, Q3 months during year 1, Q6 months during year 2. And the primary endpoint of this trial is overall survival. And we have a non-inferiority hypothesis, being that we believe in the context of brain MRI surveillance that MRI alone will be non-inferior to MRI plus PCI. So here's some notes on the trial. So it's open across the NCTN Cooperative Group Network, and it's also championed in the VA with a dedicated VA study champion. As I said, it is the first ever trial of MRI surveillance plus or minus PCI for limited stage disease and critical to resolving the conflicting data in extensive stage. There were recent closures of two trials in the small cell lung cancer space that accrued extremely well and actually closed due to successful accrual. One is NRG LU005, which was chemoradiation plus or minus atezolizumab, and the other was NRG CC003, which was PCI plus or minus hippocampal avoidance. And the closure of those trials gives us a great opportunity to bring on more sites who have an opening in the small cell and specifically the PCI space. And we invite any centers that have those trials open and are looking to activate new trials to please consider our trial. I'll have my contact information at the end. And then just a reminder that this trial allows hippocampal avoidance PCI, and that may become the key question pending the CC003 results. It was designed to be a pragmatic trial. We include limited and extensive stage. We allow any and all first-line therapies that are approved by the NCCN. We allow hippocampal avoidance and memantine. Salvage therapy is at the MD discretion, and radiosurgery is allowed. And our posture is to allow dual trial enrollments on other clinical trials wherever possible. We also have an amendment that extended the eligibility window to 16 weeks post completion of platinum-based chemotherapy, just to try to lower barriers to accrual as much as possible. As of today, we've accrued 115 patients out of a planned 600. So we are really trying to bring on new sites and to continue to encourage accrual for those that have it open. And we just want to thank everyone who's participating for helping us to enroll to this study. So from the PCI question, I'll move on to a related study and a somewhat related question. Again, looking at small cell lung cancer, but in this case, looking at patients who have already developed brain metastases. And so this is NRG CC009, and this is one of the studies that's championed here. And this is a phase 3 trial looking at stereotactic radiosurgery versus hippocampal avoidant whole brain radiation for small cell lung cancer brain metastases. The PI is Dr. Gandhi, and I serve as co-PI on this study. So brief background. For patients with small cell lung cancer, it's kind of a unique situation for those with brain metastases. Stereotactic radiosurgery has really supplanted whole brain radiation as the preferred treatment for patients with most tumor histologies, but small cell lung cancer is an important exception to that. And one of the reasons for that is that patients with small cell were excluded from the landmark trials that helped to establish radiosurgery alone for patients with limited brain metastases. And some of the reasons for that is that people have been concerned historically that small cell lung cancer patients can have short interval and sometimes diffuse CNS progression, and that if you omit whole brain radiation up front, that could potentially decrease overall survival in some of these patients. And so, but we also, on the other hand, know that whole brain radiation can be associated with toxicity to cognition and quality of life. And one of the key strategies that we use to mitigate that is by using radiosurgery up front, and then also some new strategies like hippocampal avoidance, memantine, and then sometimes CNS active systemic therapies. There is some data looking at radiosurgery for patients with small cell brain metastases, although no prospective data to my knowledge yet. But one of the largest studies was a study that we helped to lead. This is the FIRE SCLC study, and it was a large multi-institutional international retrospective study looking at first-line radiosurgery versus propensity score match group getting whole brain radiation. And in that comparison, whole brain radiation, as expected, was associated with better CNS progression outcomes, but this did not translate into an improvement in overall survival. And so this data kind of together suggests that the trade-offs associated with upfront SRS might be similar to other histologies where radiosurgery is well established. And so here's the trial schema. So this is the NRG CC009, and it's a phase 3 randomized trial of radiosurgery versus hippocampal avoidant whole brain radiation for 10 or fewer brain mets from small cell lung cancer. It's a straightforward 1-to-1 randomization. Patients with 10 or fewer mets get radiosurgery alone for hippocampal avoidance whole brain radiation 30 gray and 10 fractions. The accrual goal is 200 patients, and the primary endpoint of the study is time to cognitive failure using a standard battery of cognitive tests that have been used in other cooperative group trials. Some notes on this trial: it's the first ever, to our knowledge, randomized phase 3 trial of radiosurgery versus any whole brain in small cell lung cancer. It was activated in February of 2021. Accrual has been steady, but a little bit below expectation thus far. We're at 36, a planned 200 as of today. And just a note for logistics for folks who have the Maverick study open, patients on the MRI surveillance arm of that study who developed brain mets can co-enroll onto CC009. And so if you're interested in this trial and helping us to answer this question, we really invite you to please reach out, and we'd be happy to help try to get it open at your center. And then moving on to another champion study. This is the Alliance A071801. This is Paul Brown's study. It's a phase 3 trial of post-surgical single versus fractionated radiosurgery for resected metastatic brain tumors. And so this study actually just recently completed accrual, so we have some good news on that. The background for this study was that basically postoperative radiosurgery became the standard of care following some randomized trials showing better local control with post-op radiosurgery and then better cognitive preservation compared to whole brain. But the outcomes with single fraction radiosurgery used in those studies appeared to be suboptimal. And some other studies, non-randomized, showed that multi-fraction radiosurgery might have better local control outcomes. So this study was really trying to answer the question of what's the best radiation strategy for brain mets that have already been resected when you're treating the surgical cavity. This was the schema, straightforward randomization to single versus multi-fraction radiosurgery to the tumor bed after having a brain metastasis resected. And the status is that it would successfully completed accrual last month, 242 patients, which is really a very impressive amount for this type of study, and it accrued very quickly. And for those who have it open, Paul Brown, the PI, just asked that we remind folks that to please upload the MRI imaging, as the primary endpoint is image-based assessment of the local control. So thanks to people who have participated in this study, and we look forward to the data coming from this. And then finally, the last study that I'll present is another champion study. This is the NRG-BN009. And this is a phase 3 trial of salvage radiosurgery or salvage radiosurgery plus hippocampal avoidance whole brain radiation for the first or second distant brain relapse after upfront radiosurgery with a brain metastasis velocity of greater than 4 mets per year. So that's a mouthful, and I will try to unpack that. The principal investigators of this, again, is Vinay Gandhi, as well as Michael Chan. And so this study really is testing this concept of brain metastasis velocity, which is a pretty straightforward concept. It's basically number of new brain metastases that a patient develops divided by years. And so the higher that number is, the more brain metastases they're developing per year, and the worse their outcomes are per various lines of evidence. So here's just some evidence looking at this concept, showing that overall survival as well as neurological mortality are worse in patients who develop brain metastases more quickly. And so this raises the question of, if patients are developing lots of brain metastases very quickly, is radiosurgery the best strategy for treating them, or are these patients who would actually benefit from hippocampal avoidant whole brain radiation? And so that's the concept. All of these patients have had prior radiosurgery in order to be eligible. So they've had upfront radiosurgery, and this is at the time of relapse. So it's a salvage study at first or second distant brain relapse. And so again, phase 3 trial of salvage hippocampal avoidant whole brain radiation plus radiosurgery versus radiosurgery alone for patients with a brain metastasis velocity of greater than 4 per year. And this is the schema showing that the sample size goal is 350 patients, and primary endpoint is time to neurological mortality, and they have a rigorous definition for that. So the status of this trial was activated in December of 2020. It was endorsed by the SWOG CNS Working Group. So this trial has accrued slowly. It's at 6 of planned 200, or actually I should say 350 as of now. And so if you're interested, please consider opening and enrolling on this study. And so this is my contact and email. Please feel free to reach out to me with any questions about these trials. Thank you.
Thank you so much. Thank you so much, Chad, for that overview of all the brain met studies that you've been leading, or you've been helping us endorse for other cooperative groups. Any questions for Dr. Rastovan? Can you take the mic, actually? Because there are people on the Zoom, and if you don't mind introducing yourself as well.
Tim Robinson, Yale, CNS, heme, and inpatient palliative. For the brain met velocity study, 4 brain mets per year, which totally makes sense. But how long do you have to kind of assess that over? Do you have to kind of get at least a year's worth of, kind of salvage SRS, or at what point can you kind of, if you get 2 in 6 months, is that good enough, or how do they kind of?
Yeah, my understanding is it's so that would be like, if 6 months would be like 0.5 years, and you could use that as the denominator. So whatever your interval is, you could identify it with the time interval that you have.
So if you get it like a met 3 months later, then you're in.
Maybe, yeah.
So is this considered as an oncology study, or is this considered as a SWOG study? Because my institution is, I don't think my institution is
It's an NRG study, but I think you should have access to it. It should be open across the full NCTN cooperative group network. So by virtue of participating in any cooperative groups, I think that you would be eligible to open this.
Any other questions for Dr. Rastavan? If not, thank you so much, Chad, always for your leadership. I know you've got another meeting to go to, so thanks for spearheading us. Next on, we have Dr. Tony Wang, and can you bring up his slides, or he's going to do it over Zoom? Okay, Tony, you're on. Dr. Wang is at Columbia.
Sorry, can you hear me okay?
Yeah, we can hear you.
Okay, well, thanks again. I'm sorry I wasn't able to make it at SWOG this time around. So as someone who have been at SWOG the last couple times, I have presented this concept, and thanks to Dr. Ahluwalia for the opportunity to present my concept. This is a phase 2 study of combination atezolizumab and tocilizumab with radiotherapy in patients with newly diagnosed MGMT unmethylated glioblastoma. These are my disclosures, and they're electronically available. Some of this I suspect for those that are in the audience, and I'm sorry I can't see who's actually there. This is something that many folks who do CNS are very aware of. Glioblastoma is the most common primary brain tumor. It's most aggressive, and the standard therapy has really been radiation therapy and temozolomide. And most of us understand, although there's been some updates with respect to the WHO CNS5 classification, that a lot of the benefits primarily for temozolomide is really with the subgroup of methylated MGMT promoters. And so if you look at some of the outcomes that from the original SOUP and the paper in New England Journal of Medicine 2005, the MGMT methylation was prognostic for median overall survival, but about 2/3 of patients were unmethylated, and their median overall survival was about 12 months versus 18 months. And then in terms of the actual predictive benefit of temozolomide, with methylated MGMT, temozolomide increased the median overall survival by about 8 months. But before unmethylated MGMT, it increased the median overall survival by less than 1 month, suggesting a modest benefit with this unmethylated MGMT. Again, some of this may be we may be quite familiar with from the folks who are in the audience. So obviously, there's been a lot of new updates and data. A lot of the studies that I'm presenting are based on our colleagues from Northwestern. This is by Dr. Lamano, looking at the TCGA cohort, doing RNA sequencing and microarray data, where they demonstrated that IL-6 expression was associated with worse overall survival in gliomas. And this was published in CCR in 2019, suggesting that, again, high IL-6 expression levels, both in the RNA-seq TCGA data set as well as the microarray data, showed worsening overall survival versus those with low IL-6 expression levels. So, this is another paper that came circa around that time, revealing that, for both in the colony stimulating factor 1 and angiocrine IL-6, they induce these robust arginase 1 expression and macrophage alternative activation that are mediated through this peroxisome proliferator activator receptor dependent transcription activase. And what you're seeing here is that these anti-IL-6 therapies do reduce these M2C206 macrophage. This is another GBM mirroring model. This is a group from Penn that showed that specific knockout of the IL-6 inhibits macrophage alternative action pathways and improves survival in these GBM-bearing mice, suggesting that there may be some vascular niche-dependent mechanisms for alternative macrophage activation and cancer progression that targeting the IL-6 may offer a more selective and efficient therapeutic strategy for GBM treatments. So IL-6 signaling in GBM has been shown to reduce local and systemic myeloid-driven immunosuppression, and also enhances the immune mediator anti-tumor responses against GBM. And again, this is one of the studies that was shown from the Northwestern group. So I have shown this slide a few times at our prior SWOG meetings, but this is again experiment again done from Lamont et al. Using anti-IL-6 and NP1 for these C57BL6 mice, where they had the GL261 implantation followed by antibody treatments with either anti-IL-6, anti-PD-1, a control, or combination. And what they show here in the bottom row is both the responses on the tumors. Those are sample tumors that were actually removed, and also the actual tumor volume reduction from the type of treatments that I mentioned before. And as you can see, obviously control, you're not going to show much difference, but either in the monotherapy, but impressively in the combination treatments, the tumor volume had a more substantial response in terms of treatment, and also in terms of the percent survival, that more of these C57BL6 mice has had a longer survival time versus the monotherapy, and certainly far better than the control. So this is a group from China where they show that the blockade of IL-6 receptors by tocilizumab demonstrated a certain level of therapeutic efficacy in these xenograft models in vivo. Again, these are U251 GBM xenografts. And then, of course, again, I was just showing that, again, from the Northwestern experiments, the combined PD-1 IL-6 combination exerted these additive anti-tumor effects for these intrarenal GL261 tumor-bearing mice. So I didn't show this at the last SWOG meeting, but I do want to point out that there have been this is one currently ongoing trial led by Stephen Bagley at Penn specifically for recurrent glioblastomas, and my colleague Dr. Fabio Uemoto and I actually have had the pleasure of actually accruing quite a bit of patients on this trial using the combination of atezolizumab and tocilizumab for select patients with recurrent glioblastomas to get tezotosi followed by fractionated SRT, 8 grade times 3. And actually, we're about to enroll another patient next week for the next dose escalation. So this is the proposal that I presented before, and a lot of this was courtesy from our CNS Working Group, from a lot of colleagues who have been present here. So this is a phase 2 for newly diagnosed GBM patients with unmethylated MGMT to get, after maximal safe surgery, again, newly diagnosed GBM, to get radiation, atezolizumab, as well as tocilizumab, followed by adjuvant atezotosi. The dosing regimen is similar to what's currently being used in the NRGBN 010. Our primary endpoint is 1-year overall survival, where we expect in these unmethylated MGMT cohorts of 1 year 50% survival to expect a 70% survival. And with a 80% power of alpha 0.05, we're expecting a sample size requirement of 37. Because these are unmethylated MGMT, I think the accrual and the sample size will be short, and because of the unmethylated MGMT cohorts, the follow-up will also likely be shorter than some of our methylated cohorts. Other secondary endpoints, exploratory endpoints that we're looking for include evaluating the safety of adding the combination of atezolizumab, certainly with the ongoing BN010 combination. I think that will certainly help with our point. And we're also looking at progression-free survival, as well as quality of life measurements, and also tracking steroids use and it's associated with overall survival. So I've got one, this is about accrual, where this, this concept kind of mirrors the design of the ongoing and completed studies. So that includes Checkmate 498, as well as NRGBN 007, again, for newly diagnosed GBM that omit temozolomide, for GBM without the MGMT promoter methylation. And that accrual for those trials was not a barrier by omitting temozolomide. And that it's important to prescribe steroids only if clinically indicated. And so this is a concept that kind of follows the precedent of some other prior dual checkpoint inhibition. So follows the precedent of unmethylated MGMT only with no TMZ, no placebo, and certainly can help avoid T-cell depleting effects of temozolomide in the experimental arm. And then because of the unmethylated status, this is likely to have a shorter follow-up, as I mentioned before. So again, I want to thank Dr. Alawali and colleague at SWOGCNS, and also with the scientific team at Genentech. We're very excited initially. We thought of it as a single institution study, but thought it would be great to run it at SWOG. So thank you for that, and I'd appreciate any feedback or comment. This is my email. If you have any questions, please email me should you have any. So with that, that's the end of my presentation. And thank you again for the opportunity to present.
Thank you, Tony. Any questions for Dr. Wang? So I know there's some people in the room. Do we have any statisticians? That's what I thought. I think we spoke on Zoom. So I know, so I did present this in the rare tumor committee earlier today. The two concepts, and they were kind of well received, and they kind of mirror itself. I'll present the second one. I know there's always this talk of, should we do a straight arm phase 2 single arm study versus, I know SWOG likes the randomized phase 2. The challenge then is, I was hoping Dr. Kurzrock would be here in person so I could actually meet with her, and Ray couldn't be here. So I'll, set up a Zoom with her just to get her inference. So the thing for us is, as a small working group, we really want 2 trials to honestly go through so that Dr. Blanky can give us our committee. And it's easier to get trials through CTEP than the malignancy steering committees. And I serve on the brain malignancy steering committee, and have scars on my back, well, I've presented. So, but there's no opposition, right, to doing single arm study, especially in orphan disease state, which this one classifies, because we are even chopping up glioblastoma further. I just wanted to make sure. Okay, great. So, no. So, yeah, so it's a great thought. I think ultimately we expect this to be randomized. So the sense is, so the challenge is, and I'll tell you, serving on the brain malignancy steering committee, if you go, all we have is preclinical information. We don't have safety. We haven't shown this combination with radiation. There is some data, the trial, the NRG study is ongoing, unless that shows exciting responses. It's tough that if you go to them and they'll say, oh, we'll allow you to do a 300 or 400 patient study, because then all the resources, go forward. The other thing is, there also has to be a buy-in from the company. So Genentech needs to support it. So Tony, I'm presuming you have continued support from Roche.
Yes, so Genentech, as a matter of fact, have been with us kind of the entire way, and they initially, when we presented them as a single solution, they thought it'd be actually great to do it multi. So my understanding, and they actually want to meet with me after SWOG, would certainly be great to meet with them again.
Yeah, thanks. So Eric, to answer your question more, so when we were doing the studies through the Adult Brain Tumor Consortium, the ABTC, which was out of Hopkins, which no longer exists now because they scrapped that, and there is a U19 mechanism, which is mostly surgery drug kind of trials or very early trials. I think there's a space for doing still phase 1/very early phase 2 studies to see, at least make sure there's safety and see some early markers of efficacy. But what I'm planning to do is have a chat with Bhupendra Man, who's at CTEP, to see how open he will be, to do like more of a pre-submission call. And that's what we used to do with ABTC, and we thought that helped us succeed. And the likelihood of proposals actually getting a go-ahead is much higher if you get a pre-call with them before submission. So that's the plan to do. But there was support for this. Dr. Youngchai, oversaw the meeting today because Dr. Kazrak couldn't be there. So I think there was a support for it. I did talk to the leader from MD Anderson, Dr. Himaker, about some of the correlative work which could do and which could be supported through the cooperative group mechanism. So Tony, what I'll do is I'll probably follow up with you maybe with a call next week and with the statisticians, and then let's try to see if we can come up with the same numbers and then do a pre submission call with CTEM and then get their buy-in before we proceed further.
That sounds great. Yeah.
Maybe we could do something like we run track, which is kind of randomized phase two, but I have a number of 17 and so one and set it lower and see whether or not it's heading in that direction.
No, certainly.
I just want to
No, I certainly support that now, but the BLOB was in the recurrent GBM setting. So even if you go in the upfront setting, if your differential is really high, you'll probably need definitely excess of like 200 patients. I mean, you have to have a fairly, so in an unmethylated setting, if you really set up your control arm to be around 12 to 13 months, your experimental arm has to be over 18 months, and then you're looking at 160 patients. Which is reasonable. I mean, I'm not saying that's unreasonable, but that would be, and then but it'll go through the brain malignancy steering committee. So sometimes you go there, they're like, oh, you've not done anything. Like there is a lot of preclinical data here, so we can certainly go that route as well. So what I would probably do is do both the calculations with the statisticians and get on a call with Bhupendra Man and see which way he's supporting, because he's on the steering committees. No, so it's a great idea. But I think the numbers are a little bit more than what's BLOB, because in recurrent setting, your differential can be 3 months and your N is reasonable. In upfront setting, your differential really needs to be 6 months to have a reasonable N. For 3 months benefit, probably looking at 400 plus patient study. So thank you. Excellent questions, Eric. Thanks so much, Tony. And I did speak to Lisa. She was supportive through the radiation oncology group too.
That's great. Thanks so much.
One of these immune therapy-based trial, which is for tails, is there any kind of modification in the stats calculation? You go on the curve, it's 0, so that's
Yeah, I mean, I think, so this is going to be just an early, honestly, signal finding study. Your question really is like, for those tails, really the landmark trials, maybe OSS 2 or 3 years is more likely. But that's probably more in a definitive setting than in, here the intent is really to find a signal early on and then do a follow-up trial. Like this is just the first step to a phase 2/3, or if the results are really good, maybe a definitive phase 3. Okay, any other questions for Dr. Wang? Thanks, Tony. So can you get my slides up? Yeah. Okay, so we'll move on next to this concept that is going to mirror this one. So what approach we are taking is right now that we probably are trying to get 2 concepts in for GBM and see how successful we are with CTEP. And so this is going to be TRC-102 and temozolomide. And Dr. Wang's concept was unmethylated. This is going to be in methylated glioblastoma. And he presented good data about GBM. I don't need to tell the group about that. You have all seen the slide. We do know that the benefit with radiation and temozolomide is primarily driven by MGMT methylated patients, right? So as you can see here, if you're methylated, there's a benefit of 6 months. If you're unmethylated, your benefit is only 21 days. So this was a study that I was involved in when I was in the Adult Brain Tumor Consortium. This was ABTC 1402 study. This was TRC 102 with temozolomide in patients who had recurrent glioblastoma. So what's the basis? We know we use temozolomide. When we use temozolomide in GBM, it forms the 0,6-methyl guanine adducts, which are repaired by MGMT, but it also forms 7-methyl guanine adducts and 3-methyl adenine adducts, which are actually removed by the base excision repair pathway. And essentially, the N-methylpyrimidine DNA glycosylase, or the MPG, is the first step in the pathway of base excision repair. And this was work led by Andy Sloan, unpublished data out of Case Comprehensive Cancer Center, because TRC102 is a base excision repair inhibitor that was invented by Stan Gerson, who's the Case Comprehensive Cancer Center director. And essentially what Andy showed was that if you combine TRC102 plus temozolomide in a GBM orthopedic xenograft, you can see the beautiful curves that we are seeing in mouse models, which don't get replicated, unfortunately, in patients. So this led to this trial, which was supported by CTEP, and essentially it was a phase 2 study of bevacizumab naive first recurrence glioblastoma patients who had progressed after radiation and temozolomide. We gave everyone TRC102 and TMZ at 150 milligrams per meter square. Primary endpoint, we were told, was a response rate. So we did, to answer your question, Eric, we were allowed to do a single arm study. If you're going to do a single arm study, they like you to have response rate, especially in the recurrent setting. So here we needed a 30% response rate. In fact, FDA will give you accelerated approval if you hit 30% in GBM, because our typical response rate is 5% in recurrence. We never achieve it. So this trial, like a zillion other trials we've done in recurrent glioblastoma, unfortunately did not reach its primary endpoint. Median overall survival was 11 months, not any different than what we've seen in other studies. Median PFS was 2 months, again, very comparable to what we've seen in alkylating therapies. However, there were 2 patients on this study, which I actually had the privilege of taking care of, did really well. One of them had a progression-free survival of 18 months. The other one was 30 plus months. And we did correlative studies. Not a lot of things stood out. There was some correlation with MPG staining, but not as strong. Now, both the patients who had PFS of 11 plus months did have MPG expression. But what was more interesting was what we found was that there were, so again, small cohort, 19 patients, right? So can't make too much out of it, but 14 were unmethylated, 5 were methylated. And in the methylated patients, median overall survival was 30.2 months. Now, obviously, confidence interval is large and the lower limit is 9.5 months. So you have to take it with a grain of salt, but it was intriguing. And there was a definitely, p-value that was significant between the two, which is again, not uncommon. We see that in methylated doing well compared to unmethylated. But what was interesting was when we did, so we had tissue actually at Cleveland Clinic where we did genomic and transcriptomic analysis, and we found out that the 2 patients actually who you see in the far left, right actually, who did well, actually had a DNA damage response pathway signature. And there was a patient who did borderline well and actually had a borderline signature. He actually progressed after 6 cycles, so didn't make it to PFS 6 because cycles are 28 days and months are 30 days in a month. So based on this, we actually are proposing this study in the newly diagnosed setting. I still believe, once resistance sets in, you cannot really reverse resistance. And that was the problem with that trial, because the patients that we took were actually temozolomide refractory. And then getting a 30% response rate is like a Herculean task. So we've spoken to Charles Thier, who's the CEO of the company. The drug is actually through CTEP. All the trials have actually been done through CTEP. And Alice Chen in the CTEP has been involved with some of their colorectal studies where they've seen a nice, can you go to last time? Okay. A nice responses even in colorectal patients with the combo. So they are supportive. So what we are hoping to do is a CTEP call for both the concepts. And this one is going to be phase 1/2, but primarily is going to be looking at dose levels of TRC102, 75 milligrams per meter square to 75 milligrams flat dose to begin with, and then doing 1 dose reduction if we do not do. What we found out in TRC102 recurrent study, that 150 milligrams flat dose was quite well tolerated, but it was only given days 1 through 5. And so we did kind of some modeling, and essentially the intent is to just do it continuously, and then just mirror it with temozolomide every 5 days, because we think the two combinatorial efforts work better. So I'll reach out to our statistician. Is it authors? Yeah, and then try to make sure that we can get some numbers to send it to Bhupendra Man and the statistician at CTEP, and let's just get their preliminary input. And I'll love for you to be on the call. I will talk to Dr. Kursrog before I do that, just to get any guidance from her. And depending on what the feedback we get for single arm phase 1/2 trials versus randomized setting, we could probably go back and redo the calculations. Any questions? So it was actually, it was a PARP inhibitor. So it was actually veliparib. So it was a trial that was led by Jan Sarkaria, and the trial design was actually different. Actually, so what they did was, so Jan did xenograft models and showed that the veliparib worked better only in methylated setting. It did not work well in unmethylated setting. So where his trial design was, first 6 weeks, everyone got just radiation temozolomide, and he actually gave veliparib only in the adjuvant setting. So people got it in post radiation, and unfortunately there was no benefit. No benefit. Yeah. So it's the PARP inhibitor. There have been a couple of PARP inhibitor studies. This is a slightly different way of attacking. This is base excision repair inhibitor. And again, we want to see 36 is a small patient population, but what we're going to do is the DNA damage response pathway signature that I actually showed, it exists in around 11%. So we saw it in 2 patients out of 19. So that's roughly 2 out of 20, which is 10%. When we actually went to TCGA, we see that at around 11%. So what we're trying to do is we're only going to do it in methylated, hope that there's a 2 double hit, like MGMT is already hit because it's methylated. And then if you do the base excision repair, you're blocking all the 3 adducts the way they are repaired after temozolomide damage. So we are hoping that we might actually get a better benefit. But what we're going to do is also look at the biomarker driven to see if we can find that those are the patients who will do best. Now, obviously 36 patients, one could make a case we'll probably just have 4 patients, which is too little. Ideally would want a higher study, but again, the essence right now is, can we get these trials off the ground, show safety, and at least show preliminary hint of efficacy, whatever that is, to drive a randomized phase 2 trial. Yeah, so that's the whole intent. I feel if you have a DNA damaging approach, why would you not do it with radiation? So veliparib study, they did it later on, and I was like, there could be a census. So one of the things is the base excision repair inhibitor, at least in our hands, was less myelosuppressive. Most of the toxicity was hematological and was mostly anemia. But the problem with the PARP inhibitors are they're fairly toxic drugs, actually. So that has been the challenge. Any questions for people on the Zoom?
Man, it just dawned on me. I don't know if this has been brought up at the other cooperative groups, but this concept and my concept, we kind of had designs for some time. And as you know, there's been some updates with WHO classification, I guess. With this, for those that are like maybe the intermediate grades, but like has perhaps like a mutation that upgrades it to, would that be considered, would that be inclusive of yours? Like, do we need to update those part of the, Mamie?
Yeah, Tony, can you repeat it?
I'm sorry, I think I had my computer froze. Can you still hear me, Mamie? Okay. So I guess with the update with the WHO classification, do we need to update the concepts to reflect some of the eligibility criteria? Like, are you planning to accept all molecular GBMs?
Great question. I would do it after the CTEP call, because right now the biggest question will be what will be the concept. I wouldn't worry too much about the inclusion criteria, honestly, because some of the trials which have already started are still ongoing. We had the same challenge with one of our vaccine trials where we are actually in randomized setting. I think we can use the updated classification if the concept sees the light of the day, but that'll happen at the protocol writing stage, and then CTEP will be fine with that.
Okay.
Yeah. Any other questions? If not, then, these were the only two concepts honestly I had today. Because one of the things that I've been told is through SWOG, the preference is that they're not more than actively 3 concepts that go through a committee, just to make sure that we are utilizing our resources well. And we are a working group. We are housed under, rare tumor committee. But Dr. Kurzrock is excited about both the concepts since they are supporting each other. So we'll work on this and hopefully have updates for people maybe in a couple of months, and that's when we'll do the next meeting. But hopefully we'll accomplish the CTEP call prior to that, because I think that would be update. If people have ideas of concepts, we would love to entertain that going forward. I know Dr. Andrew Brenner has a study in brain mets though. Andrew, do you want to just tell the colleagues, and maybe in the next call you can actually present it? I know you've not formally presented it. I always ask Chad to present it, but do you want to just come and give people at least a couple minutes update on what your study is, what's the design, what's the enrollment, and what kind of, and how we can find ways to support your trial?
Yeah, that's S20.
You want to just come here so people on the Zoom can see you? They're not too many, but they're probably 5 people.
So thanks, Manmeet. That's a good opportunity here just to let everybody know about S2007, which is our study of sacituzumab govitecan and HER2 negative breast cancer brain metastases. We're currently at 15 patients out of our goal of 44. We have a preliminary analysis at about 23 patients. So it's starting to pick up a little bit, and I think we've finally got some of the institutions with some higher brain met accrual on, but there's still plenty of opportunity to participate. As you know, Trodelvy is approved for triple negative breast cancer, but no approval for brain metastases. And now we have, with the Tropics 02 study, we now know that it works for hormone receptor positive breast cancer. So we did a lot of preclinical experiments, as well as a pilot clinical trial which showed adequate levels of SN38 and responses in these patients with the breast cancer brain metastases. So we're hoping to get this completed soon so that we can get a potential change in labeling for sacituzumab govitecan to include patients with brain metastases.
Thanks so much. So what we'll do is probably we'll do our next working group meeting via Zoom, maybe in January. We'll let people enjoy the break. It'll give us enough time for the CTEP feedback. And Dr. Brenner, what I'll do is I'll ask you to present your trial so actually people can see the trial design and find ways to support. I mean, it's not a large number, but we know my thing on brain mets is they're 10 times more common than glioblastoma and 10 times more difficult to enroll to, despite being so much more common. So we've all know that it takes a long time. In fact, the NRG BN009 trial that Dr. Rastovan had showcased has been open for 2 years, and they put like 6 patients on, and they're supposed to put 350. So that's like really, that study's on life support. That's an understatement. A barrier is there are a lot of people who now think if the velocity is so high, they might just do the hippocampal, and people are just not finding it exciting enough to open. So sometimes if people believe in a concept, it's difficult to enroll in a trial that'll randomize patients, and sometimes some people find maybe it's an interesting concept, but we've struggled enrolling on that study. So it's just 6 enrollments in I think 2.5 years. Yeah, it opened in 2020, I think. Tony knows it better than me, but
Low enrollment velocity.
oh yeah, it's a really low enrollment velocity. Okay, so we kind of had a short agenda. I know it's late in the evening, so give the people time back. So thanks so much, and we look forward to catching up in January.